In this video, we will continue our discussion about the stiffness and the epithelial-to-mesenchymal transition. So here, what the researchers has done is first to check the effect of three different cell lines on different stiffness, and the stiffness they varied is using polyacrylamide-based hydrogel. And they have used the three different cell lines, that is BxPC3, ASPC1 and Suit2 and why they have selected these three cell lines is because this BxPC3 is more of epithelial in nature, where the ASPC1 is less epithelial in nature, and the Suit2 is more mesenchymal in nature. So they culture these three cell lines on all these three different stiffness of these three different stiffness of hydrogen, and then check the EMT markers. So here the results are shown for the BxPC3 immunofluorescence results are shown for the BxPC3, where we can see that with increase in the stiffness, the Vimentin signals is more concentrated, or it increases intensity, it increases, and these things they have quantified the panel B, and also the fold chain expressions are shown in this panel D also for BxPC3. The E-cadherin signals seems to be, its intensity seems to be goes down. It can be seen in the immunofluorescence also, and also in the panel D, where we can see the E-cadherins goes down compared to the low stiffness to the high stiffness, that is compared to the 1 kilopascal stiffness hydrogel to the 25 kilopascals stiffer hydrogel. And also the β-catenin, it is also one of the important markers for the EMT, where the β-catenin is localized into the nucleus. So here we can see that β-catenin's localization in the nucleus, and its quantifications is shown in this panel C, where we can see that the nucleations of the nucleus of this β-catenin is done almost in all the three cell lines. However, the fold change of this β-catenin localizations is high for the BxPC3, and for the other cell lines, these Vimentin's expressions are also gone up, but it is significantly gone up for the BxPC3. That is, the cell lines that are more epithelial in nature will show more response to the change in the stiffness towards this mesenchymal type, towards becoming the more mesenchymal type. And thereafter, one of the other important characteristics of the mesenchymal state cells is the change in the shape. So they check the shape using the roundness score, where the radii of these cells are being measured. Since the cells in the mesenchymal state are more spindle-like cells, so their radii should go down. This is what they have seen compared to the 1 kilopascals. This 4 kilopascal, 25 kilopascals roundness score goes down, and similarly for ASPC1 it goes down from 1 kilopascal to 25 kilopascals. That is statistically significant. However, for the Suit7 it does not goes down. This could be, the reasoning could be because the Suit7 are already mesenchymal in state, so they have less margin available to move into more mesenchymal state. Whereas for the BxPC3, since it is epithelial in nature, there is more margin available to convert to move from epithelial to mesenchymal state. The ASPC1 is the intermediate cell lines, so here the transitions towards mesenchymal is in between the behavior of the Suit and the BxPC3, and similar results can be explained, and other results can be explained from this similar understanding why the changes in this nuclear β-catenin is high for the BxPC3 compared to the ASPC1 or Suit2, and also these Vimentin expressions. It is, although it looks like increase, but those increases are not statistically significant. So from that they come up to the point that as the stiffness increases, cells will behave more mesenchymal in nature, and this is what could be actually happening in the cancer patients, where it has been reported that with increase in the cancer progressions, the stiffness across the cancer cells tissues is also increases. So this is what they finally concluded from this figure, that stiffness promotes epithelial to mesenchymal transition. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.